All right, so this is part 2 of the chapter 8 lecture, and we're going to talk about how microbial cells break down glucose to utilize it as an energy source. There's two components of metabolism. Enzymes are involved. The first part is catabolism, or catabolic reactions. These are the breakdown of complex organic molecules into simpler compounds, and they release energy. The second type of metabolic reaction are anabolic reactions, or anabolism. And this is the building of complex organic molecules from simpler ones. These type of reactions require energy. You will need to know the two different types of reactions. All right, and how does ATP fit into all of this? Adenosine triphosphate, or ATP, is the energy currency of a cell. It's a donor of free energy. It has 3 phosphates, and energy is stored in the phosphate bonds. Adenosine diphosphate is an acceptor of free energy. It has only 2 phosphates. So when ATP transfers its energy to something else so that cell can do work, it's converted into ADP. So is it catabolism or anabolism when ATP is broken down into ADP? Catabolism, right? Because a complex molecule is broken down into simpler pieces and energy is released. Now, what about if we were to add a phosphate back onto an ADP, turning it back into energy-rich ATP? That would be an example of anabolism. We're putting 2 things together, a phosphate and an ADP, and it's going to require energy to do that. All right, so let's talk about metabolism within a cell. We have ATP here. So it's stored energy. That energy can be used in the cell when anabolism occurs. Building blocks are put together to create macromolecules. These macromolecules are things like proteins, carbohydrates, lipids, and nucleic acids. Now, these macromolecules can be used around the cell for all kinds of different jobs. They might build cellular structures like membranes or ribosomes. They could be used for cellular processes like cell growth and division. Or they could be used to create energy storage molecules like carbohydrates and lipids. Now, when catabolism occurs, we're getting more energy here. All right, nutrients are coming into the cell, more food, right? That food gets broken down to form more ATP. Good. So anabolism is where we're taking building blocks and we're building large complex molecules. This requires energy. So ATP has to be used to do anabolism. Catabolism is when we're breaking things down, like food or glucose, into energy. So it releases energy. Every time there's an energy transfer, much of that energy is lost to the environment as heat. So whether catabolism or anabolism is occurring, it's never 100% efficient. Energy is always lost as heat whenever there's a transfer. All right, so let's talk about the role of ATP. Cells constantly generate and use ATP. ATP powers the biosynthetic reactions. There's 2 processes used by bacteria to form ATP. Bacteria utilize substrate-level phosphorylation and oxidative phosphorylation. So let's start at the beginning. Substrate-level phosphorylation is when chemical energy is released to add a phosphate to ADP. An example of this is called glycolysis. only a small amount of ATP is made by substrate level phosphorylation. Oxidative phosphorylation harvests energy from a proton motive force that adds phosphates to ADP. All right, chemical energy production. So an energy source or an electron donor. A compound is broken down by a cell to release energy. That compound could be some kind of food molecule. So for this example, we'll use glucose. So glucose is the energy source. The first step is oxidation. Oxidation is the loss of electrons. Another process that happens here is reduction, the gain of electrons. A good mnemonic device to help you remember this is oil rig. Oxidation is loss of electrons, oil. Reduction is gain of electrons, rig. So use oil rig to help you remember oxidation is the loss of electrons and reduction is the gain of electrons. All right, chemical energy. Oxidation and reduction reactions are how electrons are transferred from an electron donor to an electron acceptor. These reactions always happen simultaneously. Cells use electron carriers to carry electrons, and often is carried in hydrogen atoms. So here we have an electron donor. It's giving up its electron here to an electron acceptor. So the electron donor is giving up an electron. Is it oxidized or reduced? Good. Oxidation is lost, oil. So the electron donor is losing an electron, so it's oxidizing. And while it's oxidizing, there has to be something else that's being reduced, right? So the electron acceptor is gaining an electron, so it's being reduced. Reduction is gaining electrons. Oil rig. All right, so let's talk more about electron carriers. So some common electron carriers you'll see in this class are NAD+. It can be reduced into NADH. And also FAD can be reduced into FADH2. Electron carriers are used to power the electron transport system, which creates the proton motive force. So electron carriers transport these electrons and help build up a gradient of protons, which creates this motive force. All right, electrons are transferred to a molecule such as oxygen, which functions as a terminal electron acceptor. So we use oxygen when we do cell respiration to help take these electrons when they're done being used to generate a proton gradient. An example of this is aerobic respiration. So this is the same kind of respiration that your cells do when your cells break down food or glucose to harvest energy. All right, glucose catabolism. So microorganisms oxidize carbohydrates or sugars as their primary source of energy for anabolic reactions. Glucose is the most common energy source, and energy obtained from glucose is obtained from glucose by respiration, either aerobic respiration or anaerobic, and it can also be gained by fermentation. So some of the key metabolic pathways, we gradually oxidize glucose completely to carbon dioxide. So we break down glucose until we break it down into carbon dioxides. Glycolysis, there's a transition reaction, the Krebs cycle, and then finally the electron transport system. These are the four major pathways that make up respiration. Now, here's the equation for the oxidation of glucose. We have one molecule of glucose here, C6H12O6, and 6 oxygen molecules, yields 6 carbon dioxide gas and 6 water. Now, if you remember from high school biology, you might remember that this is a combustion reaction, right? We have a carbohydrate in the presence of oxygen, which burns and produces carbon dioxide gas and water, the 2 main components of smoke. It's the same general reaction you'd see when you burn logs on a campfire. And we know that when we burn things, it releases a lot of energy, right? When you burn a campfire, you get a lot of heat, you get a lot of light. Those are all forms of energy. When this combustion reaction happens in cells, it's more controlled than that. They don't ignite, but they do release a lot of energy. You end up with net production of 38 ATPs from the combustion of a single glucose molecule. That's a lot of energy from one glucose. Oxygen is the terminal electron acceptor here. All right, so oxidation of glucose. Glucose is a 6-carbon sugar, remember, into 2 molecules of pyruvate. So glucose is broken down into 2 3-carbon molecules. Is that anabolism or catabolism? Good, we're breaking it down and releasing energy, so it's catabolism. Good, and this is an investment phase. It does release some energy, but it also requires energy to happen. And so it actually costs the cell 2 ATPs to do this. But there's a payoff. It generates 4 ATPs, so there's a net overall gain from glycolysis of 2 ATPs. Now, for some microbes, this is the only way they have for harvesting energy. It's not very efficient, but it does allow them to extract a net gain of ATP from glucose. The overall end products of glycolysis are 2 pyruvates, so 2 of those 3-carbon sugars, 2 NADH, which are electron transporters, right? So they have reducing power, and a net of 2 ATPs. All right, so let's look at it in a visual form. We've got glucose at the top here. Count the blue carbons. 1, 2, 3, 4, 5, 6. So here's our 6-carbon glucose. We're going to apply ATP. So we're spending energy here to add this phosphate group. And chemical rearrangements occur. We add another ATP to do another phosphate addition at the end of the chain. Now, these intermediate steps it's not important that you know what happens in all the intermediate steps. It's important to understand that they occur, right? So glycolysis does cost the use of some ATP, but then that 6-carbon sugar is broken down into 2 3-carbon sugars. There's electron transfers, so NAD plus to NADH, and we're left with, at the end of it all, 2 pyruvate molecules. All right. Good. And you can also see here where ATP are produced. So each pyruvate yields 2 ATPs, so for a total of 4 ATPs. So from glycolysis of a single glucose molecule, we have a net gain of 2 ATPs and 2 pyruvates, and also 2 NADH electron transporters. All right, some microbial cells are able to move on after glycolysis to do cellular respiration. So this includes the synthesis of acetyl-CoA, the transition reactions, Krebs cycle, and the electron transport system. This can be aerobic, so it can occur with oxygen, or it can be anaerobic and occur without oxygen. And varying amounts of ATP are produced depending on what the electron acceptor is. All right, so let's talk about this transition reaction and the synthesis of acetyl-CoA. This connects glycolysis to the Krebs cycle. So it takes, as the reactants are the inputs from glycolysis, pyruvate, and the end products are 2 acetyl-CoA enzymes, 2 carbon dioxide gas, and 2 NADH electron carriers. The Krebs cycle is a cyclical process that needs to run twice for every 1 molecule of glucose. It requires input from the transition step, that acetyl-CoA, and the products of the Krebs cycle are 2 ATPs, 6 NADH electron carriers, 2 FADH2 electron carriers, and 4 carbon dioxide gas molecules. Now, here you can see the Krebs cycle. It's a cycle, so it's circular. Let's start at the top here. At 12 o'clock, there's acetyl-CoA. Now, acetyl-CoA is broken down into citrate. And again, when we're looking at the Krebs cycle, I don't want you to be overwhelmed and panicked looking at all of these intermediates. I want you to focus on what's happening with our electron transport molecules, NAD+, to NADH. I want you to see what's happening here, FADH to FADH2, so more electron transport's happening. I want you to see that we go from acetyl-CoA as a 2-carbon molecule, and then we add more things to it. It turns into a citrate. Sometimes this is called the citric acid cycle. Carbon dioxide gas is produced. ATPs are produced down here. All right. Water comes in. And then we need to rebuild. More oxaloacetate is the substance that interacts with this acetyl-CoA. So we have 4 carbons here in the oxaloacetate, plus the 2 in the acetyl-CoA come together to make a new 6-carbon molecule. All right. And it gets broken down. We lose some carbons to CO2. We are able to add more phosphates to ADP to generate more ATP. So for every 1 glucose molecule, the citric acid cycle, the Krebs cycle occurs 2 times. All right, the next step is the electron transport system. And the input for this is the reducing power from glycolysis, the transition, and those Krebs cycle reactions. Electron carriers NADH and FADH2 have reducing power. They transfer electrons to proteins in the cell membrane. The electron energy is used to eject hydrogen ions. And remember, a hydrogen ion is essentially just a proton, right? A hydrogen atom is 1 proton and 1 electron. And if that hydrogen is missing its electron, right, then all it basically is a proton. So these hydrogen ions, these protons, are pumped from inside the cell to outside the cell membrane. And that gradient of hydrogens, that buildup of hydrogens outside the cell, makes them want to move through the cell membrane badly. And that's what forms the proton motive force. When the electron carriers transfer electrons, the electron carriers are recycled. So the NADH becomes NAD+, and FADH2 becomes FAD. Aerobic respiration ultimately passes electrons to oxygen, and oxygen's the terminal electron acceptor. That's where those oxidation and reduction reactions stop. All right, so the electron transport system creates this proton motive force. The lipid bilayer, the cell membrane, is not permeable to H+ atoms. Those protons are stuck out there. They have a positive charge, and those positively charged protons build up and are concentrated immediately outside the cell membrane. Separation of charged ions creates an electrochemical gradient across the membrane, and that gradient has potential energy. Energy is harvested when the protons flow through the protein channels called ATP synthases or ATPases. Now, notice here, there's an ASE suffix on this word. What type of molecule is ATP synthase? Good, it's an enzyme. Remember, all enzymes end with ASE. So it's a special enzyme channel. And we know that enzymes are biological catalysts. And the reaction that ATP synthase catalyzes is the phosphorylation of ADP into ATP. So when those protons flow through the ATP synthase to move down the concentration gradient and through the membrane, they literally spin this. It's like a motor. The movement of those protons spins the motor in this ATP synthase that attaches phosphates to ADP and generates ATP. And this is where the bulk of the ATP are generated. Good. So here we've got our phospholipid bilayer. You can see we've got protons, H plus, being pumped out of the cell, right? So the bottom side, this is the internal part of the cell, the cytoplasm. And this is extracellular fluid out here. So this is just the outside environment, since we're talking about microbes. So these H pluses get pumped out, as the electrons are being passed down the electron transport system. And if you look, every time you see oxidation, reduction happening, these are where the electrons are flowing. See those red electrons flowing down the electron transport system? Good. So every time electrons are transferred, these positively charged protons are pumped out of the cell. So that leaves us with a huge net positive charge out here and a net negative charge in here, right? With these leftover electrons. So these positive protons really want to be in here with these negative electrons. And the only way that those positively charged protons can make it into the cell is through the ATP synthase enzyme. And as those protons come through, they turn a spinning motor pump that attaches free phosphates to adenosine diphosphate and make ATP molecules. All right, so how are 34 ATPs made from the electron transport system? Well, we get 3 ATPs for every NADH and 2 ATPs for every FADH2. So from glycolysis, let's tally them up. So we've got NADH here on the left. Glycolysis makes 2. The transition reactions make 2 NADH. The Krebs cycle produces 6. So we have a total of 10 NADH. 10 times 3 gives 30 ATP total for all 3 processes. And FADH2 aren't produced until the Krebs cycle. We get 2 of those. And those each generate 2 for a total of 4 ATPs. So we're up to 34 ATPs from a single glucose molecule. All right, so the total ATP production then for the complete oxidation of one glucose molecule in aerobic respiration. Remember, aerobic means oxygen. So this is where oxygen's the terminal electron acceptor. We get a total of 2 ATPs generated by glycolysis, 2 more by the Krebs cycle, and 34 from the electron transport system. And that leaves a total net gain of 38 ATPs generated by aerobic respiration of a single molecule of glucose. You'll want to be familiar with these numbers. They're important. All right. So aerobic respiration is where oxygen is the final electron acceptor. Aerobic bacteria will use oxygen when it's available. and a large amount of energy is produced this way relative to when other electron acceptors are used. So if oxygen is available and microbes can utilize it, they will, because it's more efficient. But oxygen isn't always available to microbes. So anaerobic respiration is when an inorganic molecule other than oxygen is used for the final electron acceptor. It's less efficient than aerobic respiration. Less ATP is generated from respiration that's not aerobic. But some other electron acceptors that are used might be nitrate, sulfate, or carbonate. All right, let's talk about fermentation. So some general fermentation characteristics are they're indifferent to oxygen. It should be one word. I'll need to correct that. Indifferent to oxygen. They can survive with or without it. Oxygen doesn't inhibit the growth of fermenters, and they don't use it as an electron acceptor. So during fermentative metabolism, organic molecules act as electron acceptors. So here's the difference between fermentation and anaerobic respiration. Anaerobic respiration uses inorganic molecules as a final electron acceptor. Fermentation uses organic molecules as electron acceptors. All right, so some of these fermentators are facultative anaerobes. Facultative anaerobes, some are obligate fermenters, so it's the only type of energy harvesting they can do. Others can switch from oxygen to other inorganic molecules as electron acceptors. So fermentation happens when cells can't completely oxidize glucose by cell respiration. Fermenters go through glycolysis. They form those pyruvic acids, those 3 carbon molecules. It generates 2 ATP and 2 NADHs. But the next steps of fermentation vary based on the organism. However, the next steps are to recycle the NADH, and carbon dioxide gas is usually a byproduct. So we've got glucose at the top of this model here. Glycolysis occurs in the cytoplasm of the cells. We've got our 2 3-carbon pyruvates here. There's water present. So NAD plus recycles. We have carbon dioxide gas that's generated. Acetaldehyde. So we break these 3 carbon molecules down. We make 2 carbon molecules. And this NADH here donates electrons and H plus to the acetaldehyde. And this helps it form an alcohol like ethanol with 2 carbons each. And that NAD plus is recycled up here. And these are the typical byproducts you'd expect to see from fermentation, right? The production of alcohol and carbon dioxide gas. All right, so these are some representative fermentation products and the organisms that produce them. I think what jumps to most people's mind is fermentation to produce alcohol, right? Like wine and beer. The organisms that do these kinds of alcoholic fermentation are Saccharomyces. It's a yeast. So we use yeast to make wine and beer. And of course, it's fizzy, right? That would be the CO2 bubbles. And we know the ethanol in there is the alcohol. But these aren't the only types of fermentation. So cheese is also made by fermentation, certain cheeses. So Swiss cheese, for example, gets its flavor profile. It's their CO2, right? Which helps to make these air bubble pockets in there. And propionic acid helps to give it the flavor. And the organism that's used to make Swiss cheese is Propionibacterium. Lactic acid fermentation is also something that's important to foods. So we have Aspergillus, which is a fungi, lactobacillus and streptococcus bacteria, and these are used to make things like cheddar cheese, yogurt, and soy sauce. All right, and then Clostridium is a bacterial organism that's used to produce acetone and isopropanol, which we use in nail polish remover and also rubbing alcohol. All right, so that's the end of chapter 8, part 2, where we discussed how bacterial cells utilize glucose as an energy source. Make sure you review the 4 different types of reactions. You should be familiar with what's produced by each of those 4 types of reactions, and you should know how much ATP is produced at each step. All right?